So, small intro about the institute. Our institute is Nanotech Research Facility, which is one of the brainchild of PSG, where we focus the entire the research in nanoscience and technology in different lines like nanometals for energy applications, sensors, fuel cells, drug delivery and biosensors, tissue engineering, water purification, and nanobiotechnology. Now, even this field has been diverse, and we have very good equipment facilities to to do research in this line, starting from TEM, and we have a very good clean rooms for making devices. We have multiple coating instruments for making nanolayer coatings, and we also have other supportive characterization facilities required for. So I'll not bore you uh, with the definition. There are different definitions which has been given for nanobiotechnology. In nutshells, it's a engineering, construction, and manipulation of entities from 1 to 100 nanometer using a biological. So there are wherever you see nanobiotechnology, you will find two type of approaches. One is from biology, another is for biology. Next one, please. So as I said, there are two aspects of nanobiotechnology. One is how you use biological systems to make a nanomaterial. Another is how a biological system itself is a nanomaterial. These two are the variations which you will see. And in with respect to biosensors, the next level, the use of biomolecules for sensing is one line. Okay. Whereas in case of making a nanomaterial, you use biological system as a catalyst or an oxidant or a reductant based on what it is, its capacity. Okay. So this is how it is. And now I will start. Hope my time is 20 minutes, right, sir? Okay. Good. So I can go a little bit slow or else I have to rush. Right. So basically, the biological components itself is a nanomaterial. Okay. Whatever we are built of thousands of nanomaterials, basically. So differentiating them is very much important, and how to use them is really challenging. So the on the broad uh, per se, there are three different classifications based on biological systems. One is protein nanostructure, DNA nanostructures, and lipid nanostructures. So which is abundant in our body system, and every organism has these three. And these are potential nanomaterials which has been not much explored. So the protein nanostructures are S-layer-based nanostructures, hydrogel scaffolds. I'll talk about this in detail. Whereas DNA nanostructures uses the ATGC base pairing capacity and how this can be tweaked to make nanostructures. Okay, and lipid nanostructures, as like today we are hearing about the emulsions. Emulsion is one of the nanostructure. So other than that, we also have liposomes, neosomes, SLNs, uh, capsules, lipid nano capsules, and nanoparticles as such. So these are different type of ones. So now we'll talk about protein nanostructures. So not only but a protein is a very good source for making nanostructures because it has a possibility of molecular self assembly. Okay, the concept of molecular self assembly is very less explored for making a nanostructure. Okay, so what is molecular self assembly? The innate capacity of protein to assemble based on either a charge of the amino acid. Okay, whatever is the amino acid you are going to use, it complementary charge. For example, if you are taking more abundant of positive charged amino acids, then you should also have a negative charge. So they couple each other, and this coupling of the positive and negative charged amino acids can be tweaked to make nanostructures. So molecular self-assembly is one that. So why this protein as a nanostructure? Because they have a lot of properties like capacity to bind metals and semiconductors and ions. So now, when a protein is having a capacity to bind to any of these metals or ions, then definitely it gives you an edge to make new materials. Okay. Now, this become a template. The proteins becomes a template which you can get rid of after making a nanomaterial. For example, you are taking an amino acid sequence, putting a oppositely charged metals on top. So since amino acid sequence is going to have a very less charge, the quantum of ions which are going to sit on that is going to be very restricted. Just washing off you get a very sleek layer of ions sitting onto a protein nanostructure. Now, if you can assemble it by ripening it, for example, for if you give it light elevation of temperature, increase in temperature, there is a process called as Oswald ripening, which happens and it makes the ions to become a nanoparticle. So you get a string of nanoparticles on the top of a protein uh, membrane or a fiber or a dot, anything. So there is a possibility to arrange nanomaterials over a protein nanostructure, which itself is a nanostructure. Okay, so nano double, you get a double layer of nano, one organic and another inorganic. Okay, immobilization of proteins to thin films. So it gives you a lot of opportunities. Protein crystallization is one more thing. So when as you purify proteins, the interaction of the inorganics are going to be very high. And it's going to be very precise also, because now you know the surface charge, not only charge, the entire composition. You can play with the composition. I'll tell you how the playing with the composition of protein is going to impact the structure of formation. Okay, and then because of this protein crystallization and the defined amino acid sequence, which you know before you start playing, you have the control over the molecular assembly. Okay, so protein structure and the amino acid sequence, the way it is being arranged determines the molecular structure of the of the nanomaterials. Okay, and it gives you a lot of possibility to make it linear. So these are some of the advantages. So what is molecular self-assembly of proteins? Molecular self-assembly of proteins basically is a spontaneous organization into a well-defined and stable macroscopic structure using a non-covalent bonds. Now, the bonds which are involved behind this is hydrogen bond, ionic bond, water, water-mediated hydrogen bonds, hydrophobic, van der Waals, etc. And sometimes they are individually involved, and most of the times they are collectively involved. Okay, so not only a single force is going to impact, but it's also going, it's collectively also the forces are going to have an impact. The best example for such molecular self-assembly is your silk protein. So if you take ஒரு silk fiber எடுத்து, if you just expand it, the concept is like one single fiber can expand up to 2 kilometer length, that much big. So you just think how much protein winding is happening inside. So you can just make nanomaterials like anything with a silk. So, how this happens? Because the molecular self-assembly. So, micelles are formed from a monomeric silk fibroin. Silk fibroin is the base of the silk, and once you start assembling it into a micelle, the micelle is further self-assembled, and then it forms a silk fiber. So, if you unwind it, it is 2 kilometers. If you compress it, it is coming into a fiber. You can see it with your naked eye. So that much is there. And what is the strength of this? They say that it's 2 billion times longer stable assembly. That means how strong it is. But that's why silk is being used in sutures. இப்போ நம்ம இப்போ wound பட்டுச்சுன்னா இப்பெல்லாம் silk suturing தான். It degrades itself. So why this is happening? Because this is the strength what it has because of the molecular self assembly. So molecular assembly gives you that strength. Okay, so this there are different type of such assemblies, type 1 to type 4. Molecular Legos, I hope there are even more advantages now, because most of these slides were made close to 16, 17, 18 during that time. Now where when this now even the protein chemistry has been expanded and a lot of much more works are there. So type 1 is a molecular Lego. It's like a normal Lego where you have a complementary sequence. So complementary sequence is like you have a plus minus, and other way it is plus on top, minus on top, and it is mixed also. You have plus minus minus. Okay. So some of the peptide which is formed like using this assembly is KFEA peptide. So they are these peptide are mostly hydrophilic, or it can also be tweaked to be hydrophobic and forms a complementary ionic bonds. Now, type 2 self-assembly is called as molecular switches. Molecular switches means you can increase or decrease the size of the peptide using an external factors like pH, temperature, or even the concentration of amino acid. Now, these peptides are very short length peptides. For example, is EAK12 peptide. what happens is they have more of aspartate and glutamate residues, which is negatively charged, and it has a lysine and arginine at the C terminus. So N terminus is rich with negative charge, and your C terminus is having more of positive charge. So this gives you the complementarity. Now you can assemble your nanostructures using this complementarity, and what happens is when you change the environmental factor like pH or temperature, the 5 nanometer beta structure of this protein changes to 2.5 nanometer. And when you bring it back to the original condition, it again expands. So this is called as molecular switches. So this expansion of nanomaterial and contraction expansion is determined or controlled factors. Okay. And the type 3 assemble is a molecular paint or molecular velcro. The importance of this is where your functionalization takes place in a proper way. You can put a protein on a substrate and make it to move front and back without detachment using such structures. So they are called as this is why they are called as molecular velcros. Molecular velcros, you know the velcro, right, which is being used in our bags and many things in textile. So molecular velcros, you can detach it but with a force, but one side is firmly attached to the textile. It's the same way. These proteins also behave in the same way. The one side of this one is a terminal segment, another is a peptide, the other end is a peptide segment, and in between you have central linkers. Now, the terminal segment and the peptides gives you attachment as well as the flexibility for these proteins to move forward and backward, and the one end is attached. Okay, so this is a type of covalent bond where you get the attachment is very strong. Okay, so therefore these structures can have a very good stability on a substance on a substrate when it is attached. So type 3 oligopeptide is uh, one of the example for such peptides. The fourth peptide is called as type 4 assembly. Is something like a surfactant where you have a hydrophobic hydrophilic interaction, and you can pack your molecules in between, and then you can make a lipid nanostructures based like that. It's a membrane basically, which is looking like a lipid, but still it is a protein. Okay. And it has a hydrophilic head composed of aspartic acid and a tail which is made up of amino acids such as alanine, valine, and leucine. So your amino acid composition, if you can, why I was talking about this earlier also, when you can change the composition of amino acids in your protein, the behavior of that and the type of assembly of the protein changes. So therefore, you can make a specific nanostructure using by changing the amino acid sequence. Now, peptide, you can easily make peptides, right? Now, as amino acid assembly is easy because now you have instruments facilities for making peptides. So you can specifically make rearrange propeptides and make a capsule. So protein capsule is possible with this. So need not be even more conventional with regularly what we do, no nano emulsions, and these are somewhat is well established protocol. Now advancing this is your peptide. So with a single protein and knowing its amino acids, you can bring this many structures. Okay, one is a microbial surface layer protein. It's a natural assembly of peptide, which happens in this one, and even a molecule like uh, now Gautam was talking about the stromata and how stromata is less involved in this particle when compared to the passive diffusion. Now take a stromata for example. If you can make a peptide which can pass through the stromatal opening, or for example if it is 5 nanometer, make a peptide which is 2 nanometer, put it along with the urea, send it into that. So stomata becomes a transport mechanism for these peptides to take urea or some specific metals. I told you this is the plus point of this one. How these structures fit into agriculture is because you have a peptide with a charge which can bind to a metal ion like a zinc or a copper or a whatever is essential elements what you want, and then you can specifically deliver into the plant system using this. So molecular nanopores is one such things where DNA has been tried. Okay, the pore facilitates DNA. So whenever there is, I'll talk about that even a little bit deeper. So when you allow a DNA molecule to pass through the pore, what happens is there is a change in electric charge because of the amino acid which is going to pass. And you can even say that what type of sequence is going inside. Okay, that much potential is there. Protein based hydrogels are very much useful in medical applications. Nano ropes, peptide scaffolds. This is one area which has been less explored in agriculture because peptides are very good ones. They are degradable, non-toxic. And one more thing is you can tweak as per your requirement. Make a patch of peptide, put it onto the onto the leaf, see how much it delivers. Peptide is going to degrade with respect to time and environmental conditions, and then it is going to release it. How the follicular takes, that's one study where we can take. And like another one is this peptide surfactants, nanospheres. All these things are potential encapsulating agents which can hold a lot of nutrients. Not only nutrients, you talk of a pesticide, you can reduce the entire content just by putting this. Now, entomologically also this is having a significance. How? Now, the smell of some, what do you call that? The original smells? Pheromones. Sorry. No, when you put a pheromone onto this, you can encapsulate a pheromone and put a patch onto the different sections in an area. Then the or outer layer full cover பண்ணி போட்டா கூட போதும். So insect coming in, repellents will be there. And if you want a positive insects to come in, that then also you can use it. So lot of potential is there in such structures how you can use it in agriculture. Nanofibers and filaments, protein arrays, bacterial adaptions, maybe this may are more close towards sensor sites. So let's explore. Then this is well studied. Making metal nanoparticles, I will not deep into this because people have studied left, right here. How a plant, they take any plant extract, grind it, take the extract, put it a little change, reduce, oxidize, then you get a nanoparticle. So it's a well studied area. Microbes are this one. Okay. This is one potential area which has not been much used. And this is called a surface layer protein. Microorganism uses this surface layer protein as a defense mechanism, basically, to get rid of its predators and environmental condition. For example, if you're throwing a bacteria onto a minor, so bacteria has to survive there. So what it develops is it coats its cell membrane using this type of proteins. The proteins are very good binders for the metal ions. So the ions doesn't penetrate into this and they still stay outside of this one. The what next happens, since it's a protein, it also has either a oxidative effect or a reductive effect which immobilizes the metal ion on top of this, so it directly prevents the entry of metal ions into the cells. So this is one area, and the assembly of this protein is very good. Like you can extract this surface layer proteins and put it onto any matrix, and uh, you can use it in sensors, you can use it in multiple things. So this is how a bacterial cell wall surface layer protein is there. The yellow ones are all. Do you have a pointer? Okay, it's fine. So the yellow ones are the surface layer protein. They are into the cell wall. So either you have to break it out to remove. You can see here the yellow one. In case of gram-negative bacteria, you have a secondary cell wall polymer. There's a small thread here. Can you see here? This secondary, if you just break it, then the surface layer protein comes out. Now, why we should remove this one? That's very important because they have the property to make a symmetry. What is a symmetry? For example, he was using a water emulsion. Now, I can use a protein, then water, then an oil emulsion. It's a hybrid. Okay, so now if you when you make a hybrid, then symmetry is very much important. Whether how the protein should be permeable, right? It should allow some molecules to pass through, or else it won't go inside. So that there comes a symmetry. So what happens is this P1, P2, P4, P3, and P6 microorganisms are evolved in such a way that the surface layer proteins are assembled as a symmetry. Okay. Now, how people have started using it? They have started using as a template basically. For example, if you see this P1, the P1, the void பாருங்க, the yellow போக வெள்ளையா இருக்கு இல்லையா? If you can see that, this is the space. Now, if you put a metal fitting that space, then remove the protein. Wherever the metal is sitting, the metal nanoparticle will take the shape of the protein. So it becomes a template basically. So you can make any type of nanoparticle, any shape of nanoparticle, and it will be defined because it is having a layer. It's an assembly basically. So it will take the structure of the protein. Okay, protein, the gap where sorry for that. Yeah. So you can make stars, you can make triangles, you can make flowers, anything like that. And it is not new, it's all already made in nature on the surface of a bacteria. So the advantage is like this units can be detached from the bacteria, assembled in suspension, put it in a solid support for like a patch. You can also make it onto a liposome surface or just allow it to float it onto a water liquid interface. Okay, liquid interface. It is a very good matrix for absorption. Okay, for example, in a conventional immobilization technique, what you have is like a you have the enzyme, the catalyst comes on top, right? In a immobilization, enzyme is immobilized, but it has to randomly travel everywhere to go into the exact pocket. So it's a lock and key basically. So it has to the enzyme and the catalyst and the substrate has to literally travel everywhere to have to go and find it. Okay. Now I'm just correlating this again with Gautam's work. You are talking about urease. You have not studied much on the urease activity. You can use this system to say, if I'm putting a urease, how your emulsion works. You need not go to the plant system itself. Just immobilize your protein urease onto a substrate like a glass substrate. Put your emulsion and see what happens. Now, then you will know how the urease actually works. Urease is available, commercially available. So you can just immobilize onto a protein and see. Onto a random, instead of random, you can arrange it on top with this type of proteins. So it gives you a defined assembly of proteins and the immobilization matrix. It's not random. So that type of advantage is there. So this I have already talked. It is a very good matrix for metal radioactive molecules. Why? Because it has this phosphate and carboxyl group. Okay. Now phosphate and carboxyl group. No, again phosphate. Phosphate is one of your major component which has to be delivered. So these proteins are rich in binding to phosphates. So just think if it can be used as a matrix to deliver, it automatically takes phosphate from okay and keeps it up for the plant use. So it prevents leaching basically. So whatever phosphates you apply, it is hold in the soil for some time when you use this type of nano encapsulation. So it's a natural one. Okay. And it has a carboxyl group also, which has affinity towards metals. So in one go, you have a phosphate and you have a cationic nature. So in one system, dual delivery, dual delivery mode. Okay. So that is possible. And it can work as a molecular sieve, ion trap, protective shell. This is what I was talking to Gautam before this lecture. How to identify or SEM TM ல எப்படி இதை பார்க்கறது? See, do you see a double layer here? Here. This is nothing but your surface layer protein which is assembled in a natural bacterial cell, Bacillus sphaericus. Now, when you go in depth with a software, with the help of a software, you can pinpointly say whether it is a I was talking about the various symmetries, like which symmetry it is. அப்படிங்கிறது வரைக்கும் போயிடலாம். And it is with the TM, if you see the scale bar at 60 nanometer, the bacteria Bacillus sphaericus has a beautiful array of this tetragonal packs. Now, when you put ions, exactly the four pillar மாதிரி நிக்கும் அந்த ions. So that's where this has the potential. That's why it is called as a molecular S layers and templates. Okay. So it's a template. Now different type of things can be brought in. So just think you have to put your imaginations to see this. Okay. Then templating is one square arrays, hexagonal arrays, oblique parallelograms, all are there. So with that, the surface layer protein ends there. So bacterial surface layer protein has enormous one. Now, this is the way, this is one of the way by which the bacteria in the agriculture soil also impacts when you put the urea on top of it. You know, one of the problem why Any bacteria in agricultural fields are resistant to not only to urea but also to uranium is because of this problem. Okay, so in Punjab sector, if you go and read the literature, excessive application of urea led to excessive uranium leaching into groundwater, which is still a problem, and other areas have been banned using urea because of this one. Okay, and it has become an environmental issue and cancer belt. It has become a cancer belt because of this. This immobilization, the organisms which are taken from that, they are resistant to the uranium because of this. Yeah, there is one report stating that the geogenic belt is also there, but 90% of the uranium which is coming out is anthropogenic. That is because of the field, because their acres is very big. No, every farmer has acres and acres, and they just randomly they are throwing urea to it. So it's they have estimated, and this uranium is coming with along with the fertilizers. You can just Google it now itself, because one of my studies that line. So Punjab belt especially has more of uranium is has become a nuisance for them now, and because of it, especially in because the level of watering and the use of urea was not controlled use. Everywhere is like that, but there it was more prominent in leaching into groundwaters. Just ஒன்றும் இல்லை பஞ்சாப் யுரேனியம் யூரியான்னு கூகுள் போட்டால் பார்த்தீங்கன்னா வந்துடும். ஓகேவா? ஸோ தட் இஸ் வேர் மைக்ரோபியல் அது அது வந்து பிளான்ட்டை மட்டும் அஃபெக்ட் பண்ணல. இட் ஆல்சோ ஹேஸ் இம்பேக்டட் தி மைக்ரோபியல் கம்யூனிட்டி தேர். ஸோ இட்ஸ் லைக் தட். ஓகே. ஸோ நவ் தி another problem, another structure with which you protein use பண்ணி பண்ணலாம் அப்படிங்கிறது filaments and fibers. So one of the example is synuclein. Basically, it's also like a ATGC based pairing. It uses oppositely charged amino acids as a row. Okay, two strands having an opposite charged amino acids, and they bind to each other because of the electrostatic complementarity, and then they form a structure. Now, you can length the length of the strands can be improved or increased or decreased based on the complementarity. So you have to put a sequence of amino acids in a peptide such that the next peptide has a complementary to that. So then you can expand it. So that is one layer. The another is nanopores. Nanopores, I told you about this. This is a small protein which is a nanometer in size, and it allows specific molecules to pass through it. Now, if you can sequence it and you can do that, for example, stomata itself, if you can sequence the protein structures of the stomata and you can make such stomatas externally and study how it reacts with any other compounds of importance, can be done externally without using a plant. Yeah, this is one example how nanopore is being used. Even ATGC base pair, when it goes through the pores, because of the charge difference, you can easily sequence it. Now, sequencing has been done with the nanopore concept. இப்போ sequences ரொம்ப சின்னதா வந்துருச்சு பார்த்துருக்கீங்களா? Handheld. Yeah. So this is how the nanopores react. You see, there's a molecule streptavidin which passes through this, and once it passes through the membrane, there is a change in the potential. Currentல பாருங்க. கரண்ட் வந்து அந்த ஒரு எந்த இடத்துல அந்த ரோப் உள்ள போகுதோ அந்த இடத்துல சேஞ்ச் இன் கரண்ட் இருக்கு. So with this, they could clearly say that which molecule is actually entering, because every molecule has its own resistance, and this resistance is calculated and seen how the molecules. For example, you have 10 molecules which is of having different type of resistance, and put on a nanopore. The preferential entry can be easily monitored. Which one enters faster? So that can be done. Okay, and then hydrogels. This is very common now. Uh, Basically, they take more of hydrophilic peptides. Okay, so they take a lot of hydrophilic peptides. They assemble it and make a gel out of it. And this gel, since it is hydrophilic, they take more of water molecules and they swell basically. So then assume if you want to deliver something, take a peptide, put along with your molecules. You can just freeze dry it and pack it. The peptide having your molecules. And then whenever you want to go to the field, unopen that, put a little bit water, it becomes wells, just put it onto the root line. So hydrogel, it still keeps water and slowly releases the drug. So you can use it in that way. Okay. So the to gel to fluid transition can be controlled by changing the pH. Basically, put it as a gel, change the pH, it liquefies and releases everything. So and it can you can control that reverse, and because it's a hydrogel, you can reverse it. So that was so far it has been studied well for drug deliveries. Nutrient delivery, I'm giving you all why I just took the structures because these are very less explored structures in agriculture. So in medical side, for sensor side, yes, too many works go on this line. But for agriculture perspective, this has not been looked into. Okay. Conventionally, we are still in SLNs, we are still in liposomes, we are still in yes. See, precision matters than the cost. For example, பெஸ்டிசைட்ஸ் எடுத்துக்கோங்க இன்னைக்கு வந்து ஒரு பழமுதிர் போங்க you can easily smell and see how much is they have dipped it into. Okay, can we prevent that by using a patch? ஒரு பெஸ்ட்டை ஒரு பேட்ச் வச்சு அதை ப்ரிவென்ட் பண்ண முடியுமா இன்செக்டிசைடு போடாம இல்லை பெஸ்டிசைடே போடாம that's where it comes in. Okay, can you repel it by using a small put it into the நீங்க பழத்திலே தொட வேணாம் லீஃபோட எட்ஜில் பின்னாடி தொட்டு அனுப்பிட்டுங்க என்டமாலஜியில் நீங்க எப்படி யூஸ் பண்றீங்க பயோ கண்ட்ரோல் same way you can use these patches without even dipping into insecticide. That's where the impact comes. That saves a lot of money in medical expenses. So you can also convert in that line, not only the production side. So, and again, these are all synthetic, easily manufacturable. Once you know the peptide sequence, you can just keep it's a production. So fibers can be easily made. Not only this, if you feel that the peptides only is impacting, we can put a nanofiber. It's going to be very less. Put the peptide on top as a patch, so the production cost will automatically come down. So basically, you have to think in a different perspective to get how to use this in agriculture is your brain, basically. Okay. So this is RADA 16, which is very famous hydrogel, peptide-based hydrogel. And பாருங்க, it starts with one sequence. Basically, this 5 nanometer, 13 nanometer sequence is the only sequence which is required. And rest of the things is just a repetition. Okay, it's a repetition of RADA sequence as a millions of thousands of peptides, and then it's a millions of peptides, and then you get a small nanogel. Okay, so that's how you are actually making this one. Yeah, nanotubes and nanospheres. This is a very good one because you have a tubular structure that may be closed. For example, it's again peptides. Use a sequence of peptides to make a tube and then close it at the both the ends. It becomes a spot. No problem. I got it. I got that. So, by stacking of peptides. So, stacking of peptides basically controls the shape and size of this one. So, how to do it? For example, you take an amino acid sequence, diphenylamine, okay, and then stack it. 1, 2, 3, 4 stack பண்ணிக்கங்க. Similarly, you take diphenyl glycine, stack it, and then use you have to just give another orientation, close it in one side. Okay. Closer of axis in one orientation, you get a tube. Right. And if you fold it like this, all the four axes, then you get a sphere. So with one alignment, you can either get a sphere or a tube. Okay. Now you can put whatever things you want inside. You can make, for example, urea or whatever is very specific to the amino acids where it can bind to that, you can take it. This I will skip because less relevance to agriculture. Traceller, okay. Yeah, this is very important. People that have studied, you would have also come across about nanoparticles in the plant earth, and it has become extinct now. If you ever try to publish something with plant extract, nanoparticle synthesis is going to fail unless and until it's a new compound itself. Okay, because people have left right extensively they have studied it because it's the easiest thing to do in nano. But there is a catch point. A plant which gives you a nanoparticle plant extract, if you take it in a different time zone, it's going to give you a different type of reaction. That has been well studied. Just an example for this bryophyllum, it's a hydrophyte. See, the takes place differently during day and night time leaf extracts. ஒரு லீஃப் எடுத்துக்கோங்க, பயோஃபிலமோட லீஃப் எடுத்துட்டு டே டயத்துல வந்து ஒரு எடுத்தீங்கன்னா it has a lot of this malic acids and I think in if I'm right, yeah, in daytime it is having more of malic acids. It's used to regenerate phosphoenol pyruvate. Okay, in night it takes malic acid from CO2 and stores it in vacuum. So the availability of malic acid itself is different during day and night, and which is actually going to have an impact on the synthesis, right? So composition of a leaf is different in day and night in the same plant, and this is very less explored. So this is one area where people actually there are reports, but not as equivalent to that of the metal other metal extracts. So redox reactions happens differently at different time zones. Not only time zone, it also depends on the nutrition condition of the plant, the how much irrigation you are giving, the water content in this one, and many more things. So such a type of diverse studies has not been done. Instead of taking just a plant, we have to think in different lines. Yeah, this tautomerization of quinones is one more area which is less explored. Okay, so the ammonia, the it's basically this plant's uptake and translocate ammonia. Okay. This is a hydrophyte basically, and it dissolves ammonia to its hydroxides. Okay, which acts as a reducing agent. And this antioxidant ascorbate, which is there, is very important, and it is involved in this reductase possibility, reductase enzyme catalysis. So this also has an impact on the nanoparticle synthesis. So I'm just skipping this because we have to study in depth to understand the possible use of such changes in plants towards a metal ion. Okay, so DNA nanostructures. I'll just stick to the main concepts. How we are using a DNA as a nanostructure is because of the ATGC base pairing. Okay, if you can assemble the base pairs according to your choice, you can make different type of nanostructures. For example, what we have made is a DNA tile. Okay, what is a DNA tile? You have two base pairs, and you know the sticky end ligation and this blend end ligation. So for this type of structures, you have to take a sticky end ligation principle. Okay, where you can make a tile first, and then with the tile, you have to attach each tile to get a cross section, and then it goes like that. So you can expand a tile. Now you can also expand like this. You can attach a gold nanoparticle to these tiles. Now here comes the sensor application. So a DNA la gold nanoparticle போடும்போது surface area will be very less, not like a conventional substrate, like you take a thin film and put a gold, surface area is going to be more. But in this case, you have a strand of DNA on which a gold is there. So sensitivity of these structures will be far ahead when compared to the conventional methods. Okay, so you can use it for sensors. This is one structure which is used for encapsulation itself. So DNA can be, you can make balls. This is taken from Nature, which was published by He et al. It's a beautiful study. If you want, you can read it. Simple base pair explore பண்ணதுதான். But if you see the methodology, it's too complicated. I read the methodology and it was too diverse. Okay. And before the assembly, you can put molecules inside. Common cages is one more structure. Lipids, you know that nanoemulsions, liposomes, neosomes, SLNs, and lipid-like nanostructures. They have this one I will skip because you are well versed with it. This, I think this is relevant to today's topic. In nanoemulsions, SLNs, solid lipid nanoparticles, equivalent to that of a bilayer nanoemulsions, what we are talking today. This is a homogeneous mixture. For example, a lipid which can be mixed with urea. I'm just taking urea as a concept. mix it with urea and make it into a solid, then solidify it. Okay. You take a lipid, mix it with urea and solidify it. Solidify it in the presence of a homogenizer, for example, or a ultrasonicator, or a compound where it will keep it in a singular form. Okay, isolated form. Then you get encapsulation. Now, if you put two lipids and do the same thing with respect to the temperature of with the different temperatures, you get layers. First you first you uh, solidify the outer layer, and then you solidify the inner layer. You have two layers inside based on the temperature deviation of the lipid. Lipid solidify basically, right? At difference in temperature. So you can use temperature as a matrix to make double layers. You can also put vice versa. How many lipids layers you can get? Multiple, right? So instead of emulsion, this is one layer where you can put molecules inside. This is how the liposomes are taken. Basically, a phagocytosis, or we call this endocytosis, opsonization are the principles by which the emulsions. Okay, so where do we use this? This is generic. You can skip it. Okay, this is from one of the review which I have taken. It can be used in sensors, pesticide delivery, fertilizers, nucleic acid delivery, and soil structures analysis. Yeah, I'll stop here. Considering time, and if any questions is there, I because very less. Yeah, it's all how you put together the same molecules, how you